Okay, so this is the centrifuge just got done spinning for the third time for the third wash. So I'm going to remove the tubes, and since this is the saline here, so what I do is I'm going to remove the saline that I put here, and then I'm going to do the same with this tube and grab a different pipette so we don't contaminate anything. So this is the saline here. So again, this is the third time that I've done this. Okay. So the next thing I do is I'm going to make a 5% solution. So what that is, it's 1 drop of red blood cells mixed with 20 drops of saline. So I'm going to get 2 more pipettes. It's lots of pipettes in this. So I'm going to, this is the recipient and this is the recipient cells. So I'm going to make sure that they match, and I'm just going to remove 1 drop of the blood here, and I'm just going to put 1 drop in the recipient cells. So it's 1 drop here. So it kind of stuck at the bottom. Okay, then it's 20 drops of saline. So I have the saline here, and it's going to be 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 18. There's 20 drops here, and I'm going to invert it like that so it's mixed. And I'm going to do the same thing with the donor. So 1 drop of the donor red blood cells. So put the 1 drop here. There we go, 1 drop. And then the 20 drops of the saline. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. So I'm going to invert these. So now I have recipient cells, donor cells, then recipient plasma and donor plasma, and then I have my control here. So I'm going to put 2 parts of plasma to 1 part of red blood cells. So I need 1 more, so I need 2 more tubes. So follow me here. And we'll get 2 more white top tubes. And I'm going to label 1 major and 1 minor. So for the major cross match, I need recipient plasma and donor cells. So recipient plasma, this one, and donor cells. So it's going to be 1 part donor cell and 2 parts plasma. So one part donor cells to 1 drop. To 2 parts plasma. Donor cells, recipient cells. So recipient plasma. One, two. So we have the major one here. Now for the minor, which is in this tube here, it's going to be the donor plasma and the recipient cells. So donor plasma, 2 parts donor plasma. One, 2. And then 1 part recipient cells. So recipient plasma, recipient cells are here, so I need to get another pipette. Oh, here's one right here. Thought I grabbed one. So just 1 drop of the recipient. Here's 1 drop. So this is for the minor. Then I'm going to have a control, which is going to be the recipient plasma and the recipient cells. So 1 part recipient cells, so 1 drop of the recipient cells to 2 parts of the recipient plasma for my control. So I have my samples here. So what I'm going to do is I'm going to incubate these for 15 minutes at room temperature, and then I'm going to look under the microscope.